Student talk today is by Sushant Potdar and it's the effect of warming on insect-pollinated plants.
Uh, Good morning, everyone. I'm Sushant Potdar, and I'll be talking on the effect of warming on insect-pollinated plants using coriander and cumin as our model species. We all know that the climate has been warming since the 20th century, and it is because of the industrial revolution that there is increase in temperature. And IPCC has also predicted that there will be a further increase of 1.5 degrees Celsius by the end of this century. And over here I'm presenting two models by IPCC, RCP 2.6 and RCP 8.5 respectively, where the first one, RCP 2.6, takes a more conservative approach by calculating the increase in temperature by taking all the mitigative actions, while RCP 8.5 takes a very non-conservative approach. Even in the most conservative approach, you can see that there will be an increase in temperature in the coming century. And what does this have to do with, how does this affect the plant-insect interaction? So warming affects both phenology and the distribution of both plants as well as pollinators. And this can have a temporal as well as a spatial mismatch between the plants and the pollinators, leading to a plant-pollinator interaction mismatch. And that has effects on the visitation number of pollinators towards the plant for pollination. And it also has the effect on the pollinators in basis of food availability and their reproductive success. Even the floral volatiles that are emitted by the flowers of the plants are affected due to warming, and that also has an effect on the plant-pollinator interaction. Hegeland et al. Mentioned that all this was happening in the temperate regions, and most of the studies are taking place in the temperate region, but we don't know what is happening in the tropics. So we wanted to know what the effects of warming are on plant-pollinator interactions in the tropics, and this is why we formulated some questions as to how plants cope up with warming in terms of growth, their reproduction, and their interaction with pollinators. And by asking these questions, we formulated our objectives to assess the effect of warming on growth, onset of flowering, floral volatiles, pollinator visitation, and seed set. We took coriander and cumin as our model species because they are mainly insect pollinated, and most of these interactions happen because of the volatiles emitted by the flowers. They are also fast growing, their lifespan is very short. So to fulfill our objectives, we set up an experiment in GKVK for 3 months. We used open top chambers. As you can see in the picture, these chambers are open from the top. They are made of polyacrylic sheets, and they trap the heat inside these chambers, and that's how the temperatures are increased by about 1.5 to 2 degrees Celsius on an average. The important point over here is that they are open from top, so the air circulation keeps happening in these chambers, and only the temperature is what it is changing and nothing else. So we had 15 pairs of OTC and control plots, and all our plants were germinated inside the plots, both coriander and cumin. We sowed the seeds of coriander and cumin in 8 plant clusters, equally dividing it into both the species. And the temperatures were recorded using a maximize I button, and CO2 was recorded using Lycos 6400. So for the first objective, the plant growth, we tagged some focal plants at the beginning, and we started taking the measurements from the 22nd day of sowing. We also measured the height of the tallest plant at the end. The onset of flowering was measured by the presence or the absence of flowering in each plant cluster in the plots. This would be per species, and we measured this from the onset of flowering till the time all the plants flowered, which is peak flowering. The floral volatiles was collected using a solid phase micro-extraction method, the SPME. So these SPMEs are these fibers that are subjected on the flowers, and the volatiles get adsorbed on them. The floral volatiles collected were analyzed using GCMS with ethyl butyrate as internal standards. The pollinator visitation was recorded by taking 2-minute scan samples, and number of each pollinators were counted. The seed set was measured after seed formation. We harvested all the plants and measured the number of seeds that were produced per plant, and we also measured the number of seeds per inflorescence for each plant. We had some predictor variables and response variables, and we took all the predictor variables as time, treatment, that is OTC or control, and the number of plants in the plots as the predictors, while we measured the response, that is the height, the flowering, pollinator numbers, and the number of seeds as response. The floral volatiles was measured using non-metric multidimensional scaling, and all these analyses were done using R. So the preliminary results is what we wanted to check whether the OTCs are working perfectly or not. So in the first graph, we can see that the temperatures on the x-axis is the medium, that is the air as well as in the soil. So both in air as well as soil, the temperatures in the OTCs are increasing by about 1.1 degrees Celsius on an average. We also see that the carbon dioxide concentrations is not changing across treatment. So we know that there is air circulation happening inside these OTCs, and the carbon dioxide concentration are similar across treatment. The plant growth was much higher in OTCs than in control for both cumin and coriander. So cumin and coriander both grew faster under warming. The x-axis is the time, the number of days across which we measured the height, while the y-axis is the height, and they both are statistically significant. The onset of flowering is measured from the time we saw first flowering till it flowered to its maximum, that is all the plants flowered. And we see that there's a clear difference in the onset of flowering in coriander, but we don't see that difference in cumin. That is to say, cumin flowered both equally in control as well as in OTCs. The floral volatiles of coriander and in OTC and control clustered separately in the NDMS analysis. We see that the responsible compounds that were used for different clustering is provided, and we can see that the concentration of these compounds is increasing in OTC plants than in control. The pollinator visitation was higher in OTC than in control. We see that Apis florea is the main pollinator of coriander, and it was much higher visited in OTCs than in controls. While Camponotus sericus, the ants, we don't know whether that's a pollinator or not, but even that was much higher in OTCs than in controls. The seed set that was formed at the end of pollination, the number of seeds produced per plant in OTCs for both coriander and cumin was much higher in OTCs than in control. And the number of seeds that were formed per inflorescence is also much higher in OTCs than in control. So this is only for coriander, where on the x-axis is the number of inflorescence per plant cluster in the plots, while on the y-axis is the number of seeds that these inflorescences produced, again per plant cluster. So as you can see that the OTCs have much higher seeds per inflorescence than the control plants in coriander. So what does all of this have to do with? Warming definitely shows a significant increase in growth, onset of flowering in coriander, the floral volatiles in coriander, as well as the seed set. So can we say that warming is increasing the productivity of the plant? Yes, so there are these positive effects of warming on plants, but there is one thing that is important is the onset of flowering can lead to a mismatch between the pollinators visiting these plants and the flowering time. This was very true in the temperate regions where the seasons are perfectly marked. While in the tropics, we also saw that the OTCs flowered, the corianders and OTCs flowered much earlier, but the pollination happened only when both the OTCs and the control flowers started flowering. So there is this area where the OTCs started much flowering, the corianders and OTCs started much flowering earlier, which is kind of a waste in case because there is so much energy that is invested by these plants in the tissues of these flowers. Even at the end, the OTCs kept on flowering, but even that is a waste because these flowers were not at all pollinated. But the knowledge of these plant-pollinator interaction in the tropics is very poorly understood. And the increase in floral volatiles as well can, due to warming, can lead to stress in plants. And this has consequences on its interaction with its pollinators. And probably this is the first study where we are looking at floral volatiles in coriander in the tropics. So yeah, so this is a starting step where we can still further research on how these interactions are different because of warming and other global changes. I would like to thank a number of people over here, mostly the Beer Lab folks, and special thanks to Harish, who mentored me in presenting this presentation. Thank you.